Welcome to today's Explainer. We are jumping right into a really fascinating topic today, the clever methods scientists use to measure the microscopic world. We're going to take a journey together, starting from the fundamental building blocks of life, like a simple, everyday onion cell, and going all the way down to the absolute mind-bending depths of the nanoscale. So, right off the bat, here is a question for you. How exactly do you measure something you can't even see? Think about it for a second. When you need to measure a piece of wood, you just grab a ruler, right? Simple. But what happens when the object you're trying to measure is a single microscopic cell? You can't exactly just slap a standard ruler down on top of it. This isn't just a biological hurdle, it's this profound mathematical and scientific problem just waiting to be solved. And the solution they came up with? It is incredibly clever. All right, Section 1, Measuring the Invisible World. Okay, let's dive into this. To actually bridge this massive gap between the world we can comfortably see with our own eyes and the world we can't, our very first and honestly our best tool isn't actually a fancy lens at all. It's just basic math. To measure a single cell, we have to zoom way, way past the centimeters and millimeters we use every single day. We literally break things down by 1,000. To put that into perspective, there are exactly 1,000 micrometers tucked inside just one single millimeter. Make sure to keep this conversion in mind because it's absolutely crucial. It's the foundation of everything we're going to do next. Basically, whenever we measure a viewing area in standard millimeters, multiplying by 1,000 is what immediately enters us into that microscopic arena. Moving right along to Section 2, the onion cell experiment. Now that we have our mathematical key, let's see how scientists actually put it to work in the lab to figure out the exact estimated size of a single onion peel cell. First, you take a transparent ruler with millimeter markings, and you place it right there on the microscope stage. You look through the eyepiece and measure the total diameter of your circular field of view in standard millimeters. Second, you convert that number into micrometers. Step 3, you take the ruler away entirely and replace it with your onion peel slide. And finally, Step 4, you simply count how many individual cells line up straight across that exact same diameter. Honestly, it's a brilliant little Sherlock Holmes-style trick. We're essentially using the known, measured size of a circular room to deduce the size of the tiny little items lined up shoulder to shoulder inside it. So, what does this all mean? Well, it gives us our formula. The estimated size of the onion peel cell is simply the total diameter of our visible field, which remember we now have in micrometers, divided by the number of cells we just counted along that diameter. It's so incredibly elegant. We didn't measure the cell directly at all. We just measured the space it occupies and divided it up. Let's actually plug in the real numbers from our source material to see this magic in action. Suppose you looked at your transparent ruler and measured a visible field diameter of 5 millimeters. We multiply that by 1,000, which means our viewing circle is 5,000 micrometers wide. Then we swap in our onion peel and we count exactly 25 cells stretching across that straight line. We take our 5,000 micrometer field, divide it by those 25 cells, and boom. We get an estimated cell size of exactly 200 micrometers per single onion cell. Just a perfect logical deduction. Which brings us to Section 3, the power of magnification. Now, calculating that a cell is 200 micrometers wide is really only half the battle, right? Because our naked eye still can't see the thing with any real detail. The light microscope makes this visible through a compounding multiplier effect. Think of it like this. A light microscope has two main lenses doing the heavy lifting. If the magnifying power of your eyepiece is 10x, and you're using an objective lens that also has a magnifying power of 10x, you just multiply them together. 10 times 10 gives us a total magnification of 100x. This means that our tiny 200 micrometer onion cell is appearing 100 times larger to our eye, finally allowing us to see its basic structure. So the crucial point is that magnification alone is just not enough. If you just blow an image up 100 times, it's just going to be a giant blurry blob. To turn the microscope into a truly powerful scientific tool, scientists had to drastically improve two other main features. First up is resolution, which is the actual measure of clarity. It's how sharp the image is. And second is contrast, which is the difference in brightness between various parts of an object. Without strong contrast, the transparent parts of a cell would literally just wash out into each other. You need magnification, resolution, and contrast, all working together in perfect harmony. All right, Section 4, the electron microscope revolution. So here is where things get really wild. Even with perfect resolution and contrast, traditional light microscopes eventually hit a hard physical limit. They can only show us so much. To bypass traditional optics entirely and truly uncover the fine details of cell structures, scientists sparked a massive new technological leap. We are basically pulling the rug out from under the light microscope to introduce the electron microscope. Instead of relying on regular beams of light passing through glass lenses, these absolute beasts of machines use a highly concentrated beam of electrons to produce their images. By swapping light for electrons, scientists totally bypass the old limitations, allowing them to reveal fine structural details that were just completely invisible before. By using electrons, we enter a staggering new realm of measurement. We're dropping all the way down from the micrometers we used for our onion cell, deep, deep down into the nanoscale. Let that sink in for a second. A nanometer is 1/1000th of a meter. 1/1000th. This is the scale where we stop looking at just the rough outline of a cell and start looking at the intricate architecture of the structures hidden within it. This level of measurement is absolutely foundational today, driving forward the advanced material science and critical medical breakthroughs that literally define modern research. And this brilliantly illustrates exactly what we're talking about. Notice the scale bar in the corner there. We are looking at details at a 0.05 millimeter scale, yet the remarkable clarity is just undeniable. You can clearly see the individual stomata, those specialized pores on the leaf, rendered with incredible texture and depth. You can see the contrast, the resolution, all made possible by the scanning electron microscope. It completely and totally transforms our understanding of biological topography. We've gone from using a simple transparent ruler and basic math to estimate a 200 micrometer onion cell, all the way to firing concentrated beams of electrons to map the alien-looking surface of a leaf at the nanometer scale. It's an astonishing journey of human ingenuity for sure, but it leaves us with one final, deeply provocative thought. If manipulating electrons is what finally allowed us to see the breathtaking nanoscale, what invisible worlds will our next great technological invention reveal? The microscopic universe is vast, and I highly suspect we've really only just scratched the surface. Thank you so much for joining me for this explainer. I hope it brought these invisible concepts to life for you.